Eduardo Kac, born July 3rd, 1962, in Brazil, is a Brazilian American artist often known for his creative intersection between art and science. He studied at the School of Communication at the Pontifical Catholic University of Rio de Janeiro and then at the School of the Art Institute of Chicago. In 2003, Kac received a doctorate from the Planetary Collegium at the University of Wales. He started his career as an artist performing in his pink mini dress as a teenager back in the 1980s. His projects then branched into the sciences, which became what he's known for today. Eduardo Kac is most known for his concept of bioart and transgenic art that use biological concepts and biotechnology for the expression of art. He is most known for his GFP Bunny exhibition, which will be discussed later. His art has been shown at the Museum of Modern Art and the Metropolitan Museum of Art in New York. As of 2025, he has exhibitions in Tate Modern London, Buffalo AKG Art Museum, and more. He has also received many awards, including the Golden Ika Award, one of the most prestigious awards in the field of media art. His art included Genesis, Agora, and the natural history of the Enigma. Let's digress a little and talk a little about me. In AB Bao, a bunch of teenagers got to experience something they've never done before, genetically modifying something. We took a gene known as GFP, sounds familiar, and somehow put it into E. coli. Now, I don't remember the exact science behind it anymore, but basically we got germs to go on a petri dish. With my somewhat deep love for biology, I want to see if there was any intersection between art and biology. Specifically, what if somebody used life as a medium for art? As I was looking into artists who were interested in both art and science, I found Eduardo Katz, who had a similar art project that seemed a little too similar to my science experiment, but still answered my question. The GFP bunny is arguably the most famous exhibition for Eduardo Katz. In the year 2000, this bunny named Elba was supposedly genetically engineered to glow in the dark. Eduardo Katz is the main idea who came up with the concept of a glow in the dark bunny. However, this project was also carried out by French geneticist Louis Marie Udevine. In a lab, the team isolated the glowing fluorescent jellyfish protein gene and implanted it into an albino rabbit's genome, and soon Elba was created in France on April 29, 2000. By using scientific knowledge, Katz and his research partner was able to bring a glowing bunny to life. He expressed his creative thoughts in the form of life. Here's a quote from Eduardo Katz that explains why he chose to use a bunny as a form of art. By creating this bunny, Eduardo Katz creates a fusion of biology and technology, using this bunny as his canvas to express his identity, thus narrowing the pre-existing gap between these two disciplines. There are not that many images on Elba. These two are really the only ones. This leads many to think, was this experiment even real? Remember this picture? We also used the GFP gene to make something glow in the dark. Instead, it's germs on a petri dish. Notice that thing underneath the petri dish? That's a UV light box. To the naked eye, the colonies of bacteria will not glow, but it will under UV light. Scientists are suspicious of the pictures of alba glowing. Once again, GFP codes for protein, not a pigment. It is unlikely that the bunny's fur will glow, but more likely its skin. It will almost be impossible for the bunny to glow like that. Though Elba, the actual bunny, cannot be found on exhibit to the general public, there has been many forms of art circulating around the concept of Elba. There are many newspaper headlines of Elba. There are also public installations in cities, and there are also other forms of art depicting Elba the bunny. In 2025, so much of the world is about AI and up and coming fascinating technology and scientific advances. However, with these new fields, there are also blurred lines between art, science, and ethics. This genetically modified bunny is a very futuristic concept that blends art with biology. His art captivates both our excitement and anxiety about scientific progress. New technology today, like CRISPR, may make it possible for scientists to genetically modify a new animal or a new plant. Today, CRISPR is being researched to edit genes to treat certain conditions like sickle cell anemia, malaria, and HIV. In fact, CRISPR is the first FDA approved treatment for sickle cell anemia. Here are some questions for you. Do you think it is ethical for artists like Eduardo Katz to create art using organisms, specifically animals, as a medium? And what are some ways of expressing the art in biology other than using a flat medium?